It's my pleasure and great honor to introduce Dr. Wayne Hendrickson from Columbia University. And he's going to tell us his reflections on resolution, revolution, and the PDB. All right, so.
I'm just trying to get shared here. Sorry about that. Where am I?
Yep, you're good.
Get to the top. Okay, thank you, Rui. It's great to be here and great to celebrate this wonderful occasion of the 50th anniversary of the Protein Data Bank. And I thought I would do a little bit of science, but mainly a reflection on my association with the PDB, which has covered the whole of the PDB. My Structure of Lamprey Hemoglobin, my thesis project, which I didn't actually finish as a thesis. I finished it after I got to ultimately to Jerry Carl's laboratory. And this paper was published in 1973, but it refers to a paper that was published in 1971. And the data that we had accumulated on the crystal structure of this protein were deposited in the first release of the PDB among the 7 that were in that release. And that result that was there was discussed at the Symposium in 1971 at Cold Spring Harbor, where the whole idea of the PDB was hatched. And the paper that we presented, Warner Love presented, was on a bloodworm hemoglobin and lamprey hemoglobin, my project of lamprey hemoglobin, and a battery of these are shown here. I don't propose to discuss these, but I did want to share with you some of the pictures. You can get them by going to the big volume that was covered there. Basically, everybody who was working in this field at that time was present at this meeting. The first protein structure was published in 1960, so this was a decade's worth of work, and many, many things had happened. But if you looked at what had happened at that meeting, all of the proteins there came from native tissues at natural abundance. The crystals were grown in batch. The X-rays were from sealed tubes or rotating anodes and mostly recorded on film. The structures were all determined by multiple isomorphous replacement, and none of them were refined.
Is from structural dynamics. He's the
Hello? None of them had been refined, and they all, the papers that were presented at that meeting and in the preceding journals, presented hypotheses very often, but almost none of these were tested. Many of the things that we normally would use to test hypotheses weren't even available. Site-directed mutation did not exist. There were natural mutants, sickle cell hemoglobin, for example, E6V, existed, and it was yet to be analyzed. Ultimately was analyzed by my thesis mentor, Warner Love, whose picture you see here with David Davies, many other people, Don Wiley and Tom Stites in this picture, Boris Strandberg, John Edsel here, and Max Perutz, really a hero of mine, Max. And here is Max, and the greatest honor of my life really was that I was invited to talk at Max's 80th birthday, which was in 1994. Don Wiley gave a talk at that meeting, and John Edsel attended. He didn't give a talk, but you saw him earlier, and 20-some years earlier, and he had aged somewhat. But he was still very vigorous at that meeting. Don Wiley died somewhat later, and there was a symposium in 2002 that at the International Union of Crystallography, which I gave a talk, and I fished out another of the pictures from that Cold Spring Harbor meeting. Don Wiley was a prominent figure at this meeting, and he wore this t-shirt that you see here, ATC ACE. He was working on aspartate transcarbamylase, and he wrote on his t-shirt, "There is no law that says things must get better." He was a little pessimistic, I guess, about his chances, but it ultimately was a solved problem, and many, many other things, of course, have been solved in the succeeding times. And that has been possible, I think, I submit to you, in large measure because there have been major revolutions in structure determination. The idea of revolutions in structure determination is on our minds because of this wonderful paper, review piece by Werner Kuhlbrand in 2014 called The Resolution Revolution, whereby electron microscopy suddenly flashed onto the scene, able to do the kinds of things we had been doing for many, many years by X-ray crystallography. And this reported that kind of work. We were already at work using EM, and I'll say something about that work in just a minute, but let me say that what was the key breakthrough that actually broke through were the direct detectors that I have in the middle here. But prior to that, Richard Hendrickson and Nigel Unwin had done 2D arrays of bacterial rhodopsin that sort of cracked open the possibility of getting to structures that weren't atomic at that first blush, but ultimately were. But it was the single particle reconstruction methods, cryopreservation, and the direct detectors all together with wonderful programs to rely on at that time that made it possible. And we adopted these work on the Ryanodine receptor, a calcium release channel. That was the work done with Andy Marks, who had been working on this, and Joachim Frank we brought in. But the key figure here was a postdoc at the time, Oliver Clark, who actually carried out much of the work, and we published these nice papers. And I just want to give you a feeling for why we were so excited about this. What we were able to get, I should have pointed out here, we have these activating ligands, calcium, ATP, and caffeine, that were all bound into this region in this red clutch. And you'll see that clutch again in a minute. Here's the density of caffeine. This is at 3.1 angstrom resolution. The protein is very large, 2.3 megadalton. And this is the core part of it. The transmembrane part is seen here. And let me go back and try to get a pointer in here. So this is the region, I won't go back to animating it, but this is the region where these ligands that are activating bind, and then it performs the trick. And we were able, in the same grids, to get multiple states. So we were able to get a closed and an open state and to refine these at the resolutions that I just indicated. And what's marvelous here is that this particular residue, this isoleucine that's at the center, when it's closed, methyl groups actually just kiss one another, sealing the pore. But when it's in the activated state, when calcium, caffeine, and ATP are present, it can open up. And it's just a marvel of that. We've seen it again here in this Bestrophin ion channel and anion channel that Aaron Auge has worked on. And here you see it closed. I don't do the animation here, but here's the big open pore and these very highly conserved, this is a pentameric arrangement of isoleucine, phenylalanine, phenylalanine that all touch in a way similar to what I just showed you for the Ryanodine receptor. So that's the transformative change in electron microscopy that we all are living with now. It's wonderful. But there have been transformative changes in X-ray crystallography as well. Preceding that and overriding all of everything is the incredible change that came with recombinant DNA expression, which happened, as I said, after the Cold Spring Harbor meeting. And it became possible to make anything you wanted, basically. And that really transformed the field probably more than any of the technology that we're talking about in structure determination. But there have been many changes there in model building, refinement methods, how we grow crystals, the way we solve structures, and also in the instrumentation, X-ray sources of synchrotrons, free electron lasers, and ever-present changes in computational procedures. These have been recorded time and again. There was the celebration of the Year of Crystallography in 2014, and at that time Nature published this catalog of milestones. They selected 25 milestones. I put red arrows here on the ones, you could go to the nature and find this so it's more readable than the image that I give you here, but 10 out of those 25 are ones that relate to biological crystallography, and I think we can all take great pride in the transformations that these milestones, these kinds of transformations. Now, I don't know what a revolution is, really, but I have arbitrarily selected 3 of these examples that I think have been really revolutionary for our science. And one of those has to do, and I'll demonstrate why I say so, one of those has to do with anomalous scattering, which is something I've had quite a bit to do with, and another is molecular replacement and the synchrotrons and FELs. And I won't say any more about those. I think we know quite a bit. I don't have a slide for that, but I do have a slide here that I made a few years ago of the transformation that's happened in how structures are determined when you're determining them from scratch completely, de novo structure determination. And what you see in red and pink are the isomorphous replacements as a fraction of all structures that were done. And in 1997, that was something, I don't know what, 83% were done by the combination of multiple and single isomorphous replacement. And at that time, 18% were done by mad phasing that we had been doing. And by the time we got to this paper in 2014, cataloging things to 2013, that had flipped. That's a revolution, when things flip. It flipped from isomorphous replacement to MAD plus SAD, single wavelength techniques, single wavelengths completely taking over, another revolution. But at this time and forward, the de novo structures are done this way. But that's a fraction of what's being done. What's being done is dominated by molecular replacement, borrowing the structures that we have in the PDB. It's totally dependent upon the PDB. And this is a picture in 1999. 20%, we did a book in which we curated these, and the curated number is 20%. The ones that people declared were 34%. That increased to 67% declared, 44% curated by my reckoning here. And by the difference is that people refer to almost everything they do as molecular replacement because they use a molecular replacement program to do something that I would call isomorphous variants. They're the same crystal over. There was no need to solve the structure again. It's just the same thing over again. It's a bit of pedantry. The fact is molecular replacement dominates everything. And given what we've learned from AlphaFold, it's quite likely that it will even more so, that it will make much of de novo structure determination probably obsolete. Remains to be seen. Next, I'd like to say something about resolution in structure determination. My title was Resolution and Revolution in regards to the PDB. So with respect to resolution, the first thing I want to say something about high resolution, something about low resolution. And the splendor of atomic resolution is magnificent. And I think we all appreciate that in this audience, and I'd like to demonstrate a couple of points about that. This is a structure, a picture painted by Irving Geis. It now belongs to the Howard Hughes Medical Institute. And it is Crambin illuminated by the sulfur atoms, which we use to illuminate the structure in the first place. And the structure is shown here at 1.5 angstrom resolution, and I'll show you better ones in a minute, but it's very nicely resolved as you see. On the left is the picture that we actually interpreted, and it's a bit of a mess apart from the sulfur atoms, which we had distinctly found, and these are the remnants of the sulfur atoms here and here for 4 out of the 6 sulfur atoms. And we noted initially this 5-membered ring, which is a prolyl side chain in the end. But this is the rub about crystallography. You have images typically that are pretty poor. We can get them to be better by improving things in various ways, a little like Francis was telling us. we evolve them, if you will. But when we get them evolved, they are truly magnificent. This is a picture of a tyrosine group. This is the oxygen. You can tell by the number of contours that it's oxygen, not carbon. Here's a peptide unit, C alpha, C, O, N, NH, that is, and C alpha, and here's a C beta. And again, the nitrogens have more rings than the oxygens than the carbons. So the overall picture is here, and I wanted to show you this advance, which is, I don't even have on my list, but depicting structures has advanced so much. And one of the great contributions is that from Jane Richardson, who drew this picture for us, depicting the beta strands that she innovated and the alpha helices that she innovated. And they are truly magnificent. You can make some sense out of this. It's hard to make sense out of these all atom pictures that are shown on the left. And this is a tiny protein, 46 amino acid residues. But resolution is not restricted to Crambin. There are many other things. This is a 1.2 angstrom resolution from an ion channel that I did with Yu-Hong Chen. It published in 2010, and we did an update by EM. This is a bacterial homologue of a plant protein called SLAC1, which we've now done as an EM structure. Not at this resolution, but it's still very good. But you here see a prolyl residue that I'm really offering you as a foil at the moment. So this structure, when you're at 1.2 angstrom resolution, it's imperative that you include the hydrogen atoms to get the refinement done correctly. You don't really see the hydrogen atoms in this density. You might convince yourself that there's a little protuberance here, that it's not clearly just the carbon, but it's very hard to see them. And in the next picture, I'm going to show you, this is 1.20 angstrom resolution. This is 1.88 angstrom resolution, a recent result from Aaron Algae on a Bestrophin ion channel that we're working on. And this now is a map from the EM, which does show the hydrogen atoms. And you can see very prominent protuberances at many of the places around these aromatic rings. Here's another one. And you can convince yourself, I think, that you would really need to do that. Why is this? And it is because the electrons are not only seeing the electrons as X-rays do. The X-rays don't really see hydrogen very well because they're very diffuse, the electron clouds, but they do have, the hydrogens do have protons, and the protons scatter electrons because of the electrical charge that brings to the nucleus. So the nuclear positions are also contributing very strongly. We didn't innovate this. This has been described in the literature, but it's a wonderful example of another side benefit, I think, of electron microscopy. At the other side of the scale is the challenge of low resolution. And I give you a couple of examples. These are artificial examples. Sperm whale myoglobin was solved and described in 1958. In 1960 at 2 angstrom resolution, full atomic detail, but at 6 angstrom resolution, it's these tubes, if you will, and we'll see more about that in a minute. And we did the same thing. We truncated our data to 5.5 angstrom resolution, solved the structure of lamprey hemoglobin that you see it here, and later on extended it to 2 angstrom resolution. Still truncated, it wasn't the full data set at that point. But you can see these tubes of density that we could make some kind of experiment about. So the question is, what do you need in order to determine a structure? So this is the result that I gave at a CCP4 workshop some years ago. I devised a term that I call the determinancy point. At what resolution is it possible to actually define a model of a given character? And the answer I get is that if I want to determine it such that I can find all the X, Y, Z's, it turns out really I also need to find the B factors perhaps ultimately, but the positional coordinates would give you X, Y, Z's, and then depending upon what the solvent content is, we have an oversampling of the lattice depending upon how sparse the protein is within the lattice, the solvent content diminishes that. So we oversample more if there's more and more sampling, so we can get to XYZs at around 3 angstrom resolution, a typical 50% solvent content. If you want to get everything at the level of finding where essentially where the backbone is, what the phi psi angles are, it turns out that if you have something like 70% solvent content, you can get everything you need in principle at about 8 angstrom resolution. So we can get by with these low resolutions, and I demonstrated that here in the structure of myohemerythrin, the first structure that I did independently. And this is the density map, again, at 5.5 angstrom resolution. It has 108 amino acid residues and 2 iron atoms in this blob in the middle here. This is a beautiful drawing made by my co-worker's wife. Keith Ward's wife did this picture for me. And then what we did, Keith and I, we put together a procedure. I guess I did most of this actually, and I'm not going to read any of this really, except to say we first fit in helices, and then we extended helices to the parts that were between the helices and at the ends of the helices by procedures that sort of combined potential functions together with density maximizations. And in the end came up with this atomic model of, well, not atomic model, the C alpha model of the protein. And you see the C alpha model on the right, superimpose that same model onto the density map that I showed you earlier on the left. And with that in tow, I then next go to Jane Richardson. She draws a beautiful picture for me following these helices the way she does them. And then after Steve Sheriff actually refined his structure at high resolution, 1.3 angstrom, it's anisotropic, 1.3 along the vertical axis here, and 1.7 angstrom approximately in the perpendicular directions. And we can now compare how well we did from this fitting at 5.5 angstrom resolution with what's the fact. And it's very, very good. As you can see, I don't actually have I don't know what the RMSD is, but you can see that it's got to be very good. Like here's residue 10, that's residue 10. Here's residue 30, that's residue 30. Here's 100, there's 100, etc. And in fact, these 18 residues in the beginning have nothing to do with helices. I was able to track them all and built that all, including a cis-proline bond in the middle of it. That's the only way to get through it. So I'm very impressed at what you can actually do at these low resolutions if you have to. And we do have to from time to time. This is work that Hao Wu did some years later. We earlier had gotten the structure of CD4 as these 2 domains, D1, D2, and we could fit those, and we did a cyanamethionine map on these. These are resolutions 3.9, 4.0, 5.0, and Hao did a great job of this. These are crystals that had been grown earlier by Peter Kwong. So we feel confident that we can really do good things, particularly if we fit domains. And I'll come back to this a little bit later. But now going back to Cryo-EM and resolution at low resolution, to my mind, 4 angstrom resolution is sort of unacceptable. I need 3 angstrom resolution, I say to myself. So we did this structure early on. This was published in 2016. I suppose it was 2015 when I got the call from Filippo Mancia, whose problem, this is called straw 6. I'm not going to even begin to describe it. And he said, we got it. And I was skeptical. I didn't quite believe it. And the model I had domain swapping. It's exotic. And this is a helix that's transmembrane, horizontal in the plane of the membrane, never been seen. So it's unprecedented, and I was skeptical. and I got Ollie Clark, who had made the map, to send it to me. And I saw the map and I was convinced. But now I had to understand it, which I did by going back to this picture that I had made many years before on one of the helices, one of the long helices in that Myohemerythrin that I showed you. And what I did with it was to just see what maps would look like at different resolutions. This is at 1.5 angstrom resolution from that atomic model. And you can see right here is a carbon and there an oxygen, a carbonyl oxygen from the helix. There's another one. They're all through it. Of course, it's a helix. It's an alpha helix, a Pauling helix. And if you degrade the data set to 2 angstrom resolution, you can still see evidence of that. At 2.5, there's a bulge. And you can imagine if you had a lot of noise in the data, it would become much more problematic. At 3 angstrom resolution, there's no bulge. It's just a helix, but you can see the spin of this alpha helix. And what was marvelous even then was in this contouring procedure that's shown here, you can feel that this at 4 angstrom resolution is a helix. You can see, you can just, and if you look at it in 3 dimensions, you really can see that it's tracing the polypeptide chain. Why? Because the rungs of this are separated well at 4 angstrom resolution. They truly are resolved because that's the spacing between the polypeptides that are in one span of the helix and the next. At 5 angstrom resolution, it's gone. It's one big tube. That's essentially the picture of, well, 6 angstrom resolution with sperm myoglobin, and that's tubes of density. This is the reason. And if you compare the two of them, they're the same, you know? So for one thing, Richard Hendrickson got it right when he came up with this golden rule of 1.43, I guess it is, as the cutoff for the contrast, the variation between split populations. Anyway, you can see the side chains are beautiful at 1.42 for this phenylalanine. This tryptophan is sticking out at you, a little hard to see it. But they're also, you can see them in the myohemerythrin one as well. All right, so for the remainder, I'm going to just go to an example, which is a problem of high current interest to me. It's an HSP70, a molecular chaperone called DNA-K in bacteria. And there are parts of this story that I want to tell you briefly that relate to high and to low resolution issues. So this is the model that we came up with to explain a lot of biochemistry data that I'm not going to tell you anything about. And it said that the structure, at the time we were doing this, there were structures of this R state that we initially had gotten by a homolog, and then my colleague Qinglian Liu and others in Manfred Meyer's group had gotten structures that looked just like this. And we had gotten individual structures of this domain and of that domain, and they were put together in an NMR analysis that got, by residual dipolar couplings, this relative orientation of the two, but they're flexibly tethered. There's no direct contact in this uncoupled state, as I call it here. And the purple thing is the linker between the nucleotide binding ATP part and the peptide binding part. And in order to explain the data that we had measured from various biochemistry experiments, we postulated that there should be another state that can now bind peptides, whereas the R state can't. And that when it binds peptide, it can hydrolyze ATP very rapidly, whereas in the absence, in the R state, the peptide hydrolysis is slow and no binding of the peptide. So that, I just give you a part of the biochemistry here. These are the data that we measured, that Qinglian actually measured. And we could fit those data and project what they would be at peptide concentrations that we couldn't reach here. And the fit is beautiful. This is the theoretical equation that I developed from this equilibrium hypothesis. It's published in this paper last year. And we've gone on to try to find those structures that we needed, we thought, in order to explain the biochemical observations. So we predicted that there must be such a structure. We built constructs that would try to stabilize it, and we ultimately got it. We first built a construct that tethered the peptide to the C-terminus of the whole thing. We got crystals that were cubic. They diffracted to 8 angstrom resolution. We couldn't solve them. So we went on and made further ideas from various things I won't tell you about, and made a construct that was truncated to this point, which is residue 540. So another 70 residues or so from the end of what we think is the right answer ultimately. And we understand why that was necessary, but you can see that the diffraction is very good. It's 1.82 angstrom resolution, and this is the model that we ended up with. But we went back with that in mind, with that evidence in hand, and Wei Wang was able to analyze those cubic crystals. They actually formed new ones, got new data, still terrible. We're up from 8 angstrom to 7.7 angstrom. And why it's bad is partly because it's complicated. There are four molecules in the asymmetric unit. There's 69% solvent in the crystal. It's also merohedrally twinned. But nevertheless, we were able to take that model that's shown here, which is a constituent of this, and to fit that as a rigid body, molecular replacement, to fit those 4 copies. And when we do the 4 copies, there are holes for this domain that you see shown in this region. We call it the alpha lid domain. The alpha lid domain fits into interstices of the lattice, and it turns out that one of them happens to make contact such that it's stabilized and into a stable conformation. That's the red one here that's fitted. The other ones are imagined. We don't know those structures. We just saw how far we could move it one direction or another within the holes of the lattice. So we believe this would be a plausible rigid body availability of this lid domain, but that it's a flexible domain. And in fact, the function, I won't go into at all, but functionally, that's critical. We need to have that. But we're very pleased to have that. However, we would like to understand many things in detail, the enzymatic reaction in particular. And so we made just the nucleotide binding domain with the information that we had from these new structures. We were able to include a piece of the peptide that could make it be able to bind ATP. So we get this ATP structure. ATP is hydrolyzed by this protein. So we had to do something, and we did that by mutating this residue. It was a residue that others had discovered as being important for the hydrolysis reaction. It doesn't kill the reaction to make that threonine into an alanine. You just see the C beta atom here. But with that mutation, it now is sufficiently slow in hydrolysis that we can crystallize it at 4 degrees. And we get this structure that's at 1.44 angstrom resolution. And we are able now to take that crystal, we make a new crystal that's crystallized with calcium, but with a serine replacement. So serine is very much like threonine. It hydrolyzes very fast if we have magnesium, but if we have calcium, it blocks the reaction for reasons that we now understand from having that structure. But if we now soak that in magnesium, we can get the reaction to go in cristallo. It's a process that Wei Yang had done a few years before, so we're just copying her. And we can carry out those reactions and quench reaction products to get steps along the way. When we do that reaction, replacing calcium by magnesium, in the case of the serine, at 4 degrees, it stalls partway along the line, and we get this what we call the ground state. It's gone to a prime state, and in this prime state, a water has come in where there was no water before in the pink one, but in the the green colored carbons now are moved, the NH hydrogen bonds, to that water molecule that turns out later to be critical for the reaction. And I'm just going to take you through a couple of steps of the reaction that we've seen by doing essentially a time resolved study of the reaction in the case that's shown here of the working from the T199S protein. We've also done this with the wild type, and we get really fantastic results that I don't have time to tell you about, but I'll just show you this. What you see here is the ground state, that's like in the alanine mutation, and this is water W1. And if we now go to the prime state that I showed you a minute ago, what has happened is that this water that we call W4 has come in, and now hydrogen bonds both to these nitrogens in this glycine loop and also to the gamma phosphate, putting it into a position where the reaction can take place. And now I want to call your attention to this water, which in the next step moves from there to here. And there it's in line for hydrolytic attack. And there's much more to the story. I'm just going to give you one more picture, and that's the end. And that is, after having cleaved, it now is in, we see the phosphate here. Now, the truth is we're farther along in the analysis, and we're doing a lot of quantum mechanical QMMM calculations on it as well. And it's not a phosphate, it's a phosphite at this juncture. So there's much more. So stay tuned for the future, but that's the end of my story. It leaves me with but one thing to do, two things to do. First of all, happy birthday, PDB, and what a great half century it has been. And the second thing is to say thank you to many, many people. I think I've included the people that I've mentioned today. I won't repeat their names, but hats off to Warner Love, who started all of this, and hats off particularly to Wei Wang, whose story I told you at the end. That concludes my remark. Thank you, Marie.
Thank you. Thank you for a fascinating talk. So I saw there's questions on chat. James, do you want to unmute and ask your question?
Sure, yeah. Hey, Wayne, James Holton here.
Hi, James.
Something that I've been wondering about for a while, I wonder if you had an insight, was the maps looking better from EM. How much does that have to do, you think, with the sharpening? Because sharpening is sort of intrinsic to EM, but it's not something we normally do with X-ray maps. But if you can always make a map look better by sharpening it, as long as the signal to noise is good enough. So what role do you think that's playing?
Well, that's a good observation, and I don't know. I certainly haven't done any comparative studies myself, so I can't really describe it. I just want to point out, though, that whether, so perhaps we could make more attractive maps if we did them sharpened typically for crystallography. I haven't compared that, but I think, for example, I gave you this map that showed what I advertised to being showing evidence of the hydrogen atoms because of the nuclear scattering. And I think that we would never get these protuberances that I showed you for the hydrogens there by any amount of sharpening on the X-ray map. I may be wrong about that. I mean, there are little tips, I want to say, off the carbons, and perhaps if you sharpen, they would be much more pronounced. I don't know.
Okay.
It's a
Thank you.
Any other questions? Does anyone else have questions? Trying to see if there's any raised hand. All right, so if no further question. Oh, Stephen, yes.
Ray. Wonderful talk, Wayne. Thank you very much from the RCSB Protein Data Bank for contributing to this celebration. I wondered if you wanted to speculate at all on the further on the impact of AlphaFold as it's going to relate, or Rosetta fold for that matter, as they may relate to lower resolution X-ray and lower resolution EM studies.
Yes, and this has been commented on by others, and I can only echo the observation that, well, I also made an observation earlier. I think it's very likely that that it will make a lot of the de novo structure determination things, the nominal scattering work of that kind, essentially obsolete, because if it may not, there may be still problems, but many of the, we know that if you get a toehold by molecular replacement, we can go a long way toward completing the job, and even fragmentary pieces that are well folded and calculate well by the predictions from AlphaFold or other AI approaches, I think will be enough to solve many, many structures. As an example, we did a structure a few years ago of a membrane protein, published it, and I got a call from Randy Reed, or an email from Randy Reed saying, I was able to solve your structure by one helix. He took one alpha helix and he solved the whole thing.
Amazing. Amazing.
Using our data from the PDB.
Right. So what about construct design?
Construct design is I think
problems.
First impact will be on construct design. We're actively doing this now in our membrane protein consortium compo, and we're very optimistic. We don't really have results yet, but we're optimistic that we can do, comparative analyses of design construct.
Yeah, great. Thank you very much. And thank you again. Spectacular talk.